Good morning, students. This is Dr.A.Nirmala, Associate Professor of Zoology. Today, we are going to see on the topic morphogenetic movement. What is meant by morphogenetic movement? Movement of cells from one place to the another place to bring a new shape to the embryo is called as the morphogenetic movement. During gastrulation, the blastomeres move from one place to the another place. Their cells movements transform the blastula into the gastrula. These movements leads to the formation of new shape to the embryo. This movement is called as the morphogenetic movement. The movement of cells from one place to the another place is a common phenomenon in the embryos. These movements are of two types. They are epiboly movement and emboly movement. Epiboly movement is called as the movement of cells on the surface of the embryo. During gastrulation, the ectoderm spread anterior-posteriorly as well as in the laterally. As a result, the entire gastrula is gradually covered over by the ectoderm. The neuroectoderm extends anterior-posteriorly as a narrow strip of cells. Next one is the emboly movement. Emboly movement is the movement of cells from surface to the interior of the embryo. The emboly movement occurs in various types. Let's see one by one. They are convergence, involution, divergence, invagination, polyinvagination, ingression, delamination, congrescence, infiltration, constriction, extension or elongation. Let's see convergence. Convergence is the movement of cells towards a single point. In amphiaxis and frog, the endoderm, mesoderm, and notochordal cells on the surface of the early gastrula migrate towards a single point of the blastopore. But in chick and mammal, the cells from the surface of the blastomere move towards a primitive streak. Next one is the involution. During involution, the cells are rolling in. That is, the cells located on the blastoporal lip move over the lip and brought to the inner side of the lip. Similarly, the cells of the blastomere and primitive streak tend to turn a long and a primitive like structure. Next one is the divergence. Divergence is the antithesis of convergence. In divergence, the cells move in different direction from a single point. The migration of involuted cells from the blastopore or primitive streak of the interior of the embryo is the good example. They migrate in different direction from the blastopore to the primitive streak. Next one is the invagination. Invagination is the infolding or inpushing of cells into the blastocele. The process is the formation of a cavity that is archenteron, and the archenteron is surrounded by a cells in the folded form. Next one is the polyinvagination. Polyinvagination resembles the invagination, but here the outer layer invaginate in many places. The individual cells or group of cells are involved in this process. This process occurs during the formation of endoderm in amniotes. Next one is the ingression. Ingression occurs in the embryos of reptiles, birds, and mammals. During this process, individual cells or group of cells separated from the primitive stick and migrate into the space between the epiblast and hypoblast. Next one is the delamination. Delamination is the separation of layer of cells from another layer by splitting. The separation of endoderm, mesoderm and notochordal cells from each other is the good example of delamination. Next one is the congrescence. This is the migration of group of cells towards each other until they meet and fuse together form a single mass of tissue. This process occurs during the development of feathers in birds. Next one is the infiltration process. In this process, development of endoderm in amniotes, the individual cells detach themselves from the surface of the blastoderm and fall into the primary blastocell. Here, they arrange themselves to form a layer of endodermal cells. Next one is the constriction. Constriction is the reduction in the size of blastopore. The blastopore is very small but soon the lip of the blastopore begin to contract and the opening becomes smaller and smaller in size and even gradually they reduce and closed. The last one is the extension or elongation. It is refers to the anterior posterior elongation of the part of the embryo. Example is the neural plate and notochord is the best example of extension or elongation. Coming to the next is the mechanism of morphogenetic movement. The mechanism of morphogenetic movement of mesenchyme cells in sea urchin is first studied using cinematography by Gustafson and Kinada in the year 1956. During gastrulation, the inner end of cells at the vegetal pole bulge out and develop many pseudopodia-like structure. This results in the change of the shape of the cells. As the inner cells of these cells are expanded and protruded in the vegetal pole, drawn inwards. This caused the formation of orchentron. As the invagination is going on, the blastula is gradually converted into double-walled gastrula. The mesenchyme cells formed from the invaginating endoderm, the cells started bubbling on the inner surface and they producing rounded protrusion like structure, and then the cells are detached from the adjacent cells and they are released into the blastocell, and then the blastocell become a spherical in shape and they send out a slender filamentous pseudopodia towards the inner surface of the ectoderm, and the pseudopodia come in contact with the ectoderm and they are attached firmly. Then the pseudopodia contract and the results, the mesenchyme cells are loosened and drawn towards the inner surface of the ectoderm. In amphibian, gastrula is initiated by the appearance of a groove just below the equator. This groove represents the blastopore and it is produced by the invagination of the endoderm and mesoderm cells. The cells undergo tremendous changes before they are starting invaginating. The inner end of the cells expanded and outer end contracted. As a result, the cells attain a bottle-shaped structure. The neck of the bottle gets attached to the surface and the body cells is drawn away from the surface. The neck region of the bottle-shaped cells firmly held together by cementing substance and the cells in the blastopore region moves inwards. The The surface of the embryo in this region is pulled inward. This result is the formation of packet-like rudimentation of the organteron. This is about the mechanism of morphogenetic movement. Thank you, students. We will see in the next chapter. Thank you.